but that I really wanted him to speak for this community. And so he was gracious enough not only to come give a talk, but to do it on relatively short notice. We had an opening. So thank you so much, Koichi, for doing that. We really appreciate it. So Dr. Takahashi did his clinical training actually in Japan before coming to MD Anderson to do fellowship work. And then stayed on. He was promoted to an assistant professor in 2014 and has really a very interesting history. I'm not sure I've ever seen this before. He was hired in 2014 based on his clinical credentials, then did his PhD as an assistant professor and was awarded his PhD in 2020. So really challenging way to go, but boy, has he done well. So essentially his PhD thesis is essentially what got him promoted to associate professor in 21, I would suspect. So a really interesting path. But in any event, as you can tell from his title, his focus has been around essentially clonal dissection of AML. And one of the reasons I was so excited to bring him here is that the work that he's doing really goes way beyond just AML. I obviously mentioned AML, but for anybody listening, the tools that he's brought to bear are equally relevant to any form of tumor biology. So we all spend a lot of time, or have spent a lot of time for quite a while now, talking about tumor heterogeneity. But the tools to really dissect tumors and really understand this heterogeneity are really quite new. You're all aware of these many single-cell omics platforms that have sort of come online in the past few years. And what Koichi has done that's really exciting, and he's going to show you this data today, is taken sort of the latest, greatest single-cell platforms to do the genetic dissection of tumor pathogenesis. And the results are really quite beautiful, and I think, again, have ramifications for many forms of cancer. So without any further ado, Koichi, again, thank you so much for being here today.
Thank you so much, Greg. Really, thanks for the nice introduction. And yeah, we just met maybe like 2 or 3 weeks ago, and it's so nice to have an opportunity to also present in this seminar at the University of Colorado. I wish I was able to be there, of course. Houston is still quite humid, even though it's October, so I really wanted to get a little more cooler air in Colorado, but nonetheless, maybe we will have some opportunity in the future. But as Greg mentioned, today's my topic is to try to dissect this clonal heterogeneity, or in other words, intratumor heterogeneity in acute myeloid leukemia, and how those information helps us understanding the biology of AML, as well as clinical treatment in AML. This is my disclosure. And I don't think today's talk has any relevance to my relationship with the pharmaceutical companies that's listed here. So just a quick outline today is I'm going to go over briefly about the clonal heterogeneity in overall cancer and also in acute myeloid leukemia, although I know this is not really needed for this audience. And also next, we will go through some of the methodological advancement in measuring these clonal heterogeneity, especially the single cell DNA sequencing technologies that is becoming more and more available recent years. And the biological implications of understanding these clonal heterogeneity, as well as clinical implications of knowing these clonal heterogeneity and evolution patterns in acute myeloid leukemia. So again, I mean, this is really not a necessary slide for this audience, but as everyone is aware, AML is an aggressive blood hematologic malignancy that is driven by this leukemia stem cell that has increased self-renewal and decreased differentiation capabilities. And this, I know this is this oversimplified concept of how AML develops, but nonetheless, this is kind of the global understanding of how AML develops. And the recent genomic studies using next generation sequencing really helped understanding or the dissecting this heterogeneity among the patient. So what we call this interpatient or intertumor heterogeneity, that although everyone has the same diagnosis of acute myeloid leukemia, their genetic features is vastly different among each individuals. And depending on what combinations of mutation you have or what mutation you have, we are now in the era where we can recommend tailored or the precision treatment against what genetic mutation you have. For example, here, what I'm listed here is this IDH inhibitor for IDH mutations or FLT3 inhibitor for FLT3 mutation, or the most exciting things recently is this menin inhibitor against MLL rearrangement. And using the wide targeted panel sequencing technologies, Papa-Manuel group have said that, found that, at least approximately 117 driver genes are in play in acute myeloid leukemia, and probably more if you use a much wider sequencing panel. However, at least there's roughly 117 driver genes in play, and only the fraction of them are at least actionable at this stage. So there will be more and more efforts to be done in order to identify and develop the targeted therapies against each of these driver genetic events. So this is the interpatient heterogeneity that's being increasingly revealed by these next generation sequencing effort. Today's my topic is going into intratumor heterogeneity. So dissecting the heterogeneity or the individual difference of each leukemia cell that is inside the one individual patient. So a lot of work already has really already shed light on the genetic heterogeneity among each individual leukemia cells, or even the phenotypic or the morphological heterogeneity among each leukemia cell. Although each leukemia individual cells looks similar, there is slight differences in the morphology, and those things have been already published and reported based on the earlier work. And in the leukemia field, this spatial heterogeneity is something that we don't pay so much attention to it, but in the solid tumor field, this technology to dissect the spatial heterogeneity and the impact that has on the clinical treatment, as well as the biology of the tumor, is increasingly recognized. And I'm pretty sure in the leukemia field too, that the spatial heterogeneity might also get into play soon. And why do we need to really dissect or understand this intratumor or clonal heterogeneity better in not only the cancer, but also in the acute myeloid leukemia? And that's because our tumor ecosystem itself is a mixture of heterogeneous cell population. It's actually not consisted only by the leukemia or the cancer cells, but it's a mixture of a lot of mesenchymal cells or supporting cells. And understanding this heterogeneity of the tumor ecosystem itself has strong clinical and also biological implications. And also heterogeneity, like the species heterogeneity, it is a consequence of how the evolution took place. So if you have a better picture of the genetic heterogeneity in your sample, you also have, you're also able to infer some of the evolutionary patterns better. So actually from that perspective, describing the tumor heterogeneity or the clonal architecture in as much as you can in a precise way, that allows us to infer the clonal evolution pattern well. And it also has a clinical implications, as everyone already is aware, that clonal heterogeneity is a major driver of the therapeutic resistance. In some case, also a clonal heterogeneity is also a driver for the poor clinical prognosis in some tumor types. So from these reasons, I think understanding the intratumor heterogeneity or the clonal heterogeneity better has lots of clinical and also the biological implication. So what is the tool, what kind of tool do we have to measure or accurately describe the intratumor heterogeneity? Historically, we've been using a microscope to morphologically assess the heterogeneity among each cells, just by how it looks like or how the morphology looks like. Flow cytometry is a single cell tool to analyze the heterogeneity of the cell surface marker patterns. And this is, as everyone knows, it's been used for diagnostic purpose, MRD purpose, has lots of clinical utility. And most recently, we are also relying on this genetic or molecular platform to dissect this genetic or molecular heterogeneity that can be partially done by the bulk sequencing using the next generation sequencer. And for the past few years, we are also increasingly relying on this single cell platform to dissect the clonal heterogeneity. Much of the work in the past few years, understanding the intra-tumor heterogeneity or the clonal heterogeneity has been relying on this bulk next generation sequencing data. So when you have a leukemia samples or cancer cell sample in your front of you, you smash this, extract the DNA and mix together in a one Eppendorf tube and run NGS, and you get this information about the variant allele frequency. For example, in your AML, if you have 4 different mutations, you have this types of variant allele frequency. And there is a various computational tools that will help you deconvolute the clonal architecture based on these variant allele frequency information, such as PyClone or Cyclone or Expanse. And these computational algorithm helps really you to understand roughly how many clones are in play for your samples. And you can measure this, the level of intraclonal heterogeneity based on these clone numbers. And you essentially use this hierarchical system of variant allele frequency and you group them with the similar variant allele frequency and estimate the number of clone size or the subclone numbers. But these computational algorithms can do so much because you are essentially smashing all the samples in the beginning. And during this process, essentially, you're losing the individual cell information. So even though these computational algorithms are sophisticated enough, you cannot really dissect the subclonal architecture if you only have this bulk sequencing data, especially with the one time point samples. And later on, I'm going to talk a little bit about if you have a multi time point or the multi-site samples, even with the bulk sequencing, dissecting the subclonal architecture can be precise. However, oftentimes we are only relying on the one-time biopsy sample. So if you only have a one-time sample with the bulk sequencing data, it is extremely difficult to dissect the subclonal architecture, especially understanding the branching subclonal architecture like this. So the idea is, why don't we just sequence individual cells one by one and analyze the clonal architecture based on the single cell genotype information? An idea. This sounds very, very simple. I think there has been a technological hurdle that is hindering these single cell DNA sequencing in the field. But nonetheless, a lot of investigators across the world have tried to perform this single cell genotyping effort in acute myeloid leukemia using a conventional method. So essentially these studies had relied on this flow cytometry using the flow cytometry to essentially isolate the single cell using the flow cytometry and amplify the genome within that individual cells and perform the whole genome sequencing or the amplicon sequencing to understand the single cell level genotype information. Obviously, because using flow cytometry to isolate single cell is a labor-intensive process. So because of this, the number of cells, the number of total cells you can analyze or you can genotype has been limited to the degree of like 50 cells or 100 cells. And also the number of genes that you were able to interrogate has been also limited unless you perform this whole genome amplification process. And because of this unique kind of limitation in the single cell DNA sequencing field, or the single cell DNA sequencing has been associated with this unique kind of the limitation of difficulty in isolating individual cells. Again, previous study has been relying on this flow cytometry to isolate each single cell, which is labor intensive and as a result, low throughput. And also each single cell just have too little amount of DNA to analyze the DNA directly in the sequencer. So there is always this process of whole genome amplification that is needed, but that has been associated with various false positive mutations or the false negatives, which is called like allele dropout. And there has been a various method of whole genome amplification, which has its own unique false positive and the false negative limitation. And this is nicely reviewed in this Nick Navian's reviewed in the Molecular Cell in 2015. And also, there is another unique limitation for the single-cell DNA sequencing, which is accessibility problem. DNA is tightly compacted inside the nucleus, and going to that nucleus at the single-cell level has been technically very challenging. Unlike the RNA, which can be in the cytoplasms, and also there have been multiple copies inside the cytoplasm. So because of this kind of unique limitation at the single-cell field, single-cell DNA sequencing development has been lagging behind the single-cell RNA sequencing technology. So it is actually not too surprising that single-cell RNA sequencing platform like a 10x Genomics has been commercialized earlier than the single-cell DNA sequencing platform that I'm going to talk about a little bit more. So since 2016, we were fortunate to have a collaboration with the company called Mission Bio, located in the San Francisco, who were developing the platform to enable this high throughput single cell DNA sequencing. So we had a pilot experiment together using our primary AML patient samples, and we used their beta version kind of the machine. And we were very surprised initially with the performance of this machine, because they were able to genotype roughly 5,000 to 10,000 cells in one run, which is remarkably higher than the throughput that we were getting from the conventional methods. And this platform does not involve the whole genome amplification process, although it does have an amplicon amplification process. And therefore, there was this low allele dropout and also low false positive mutation associated with this platform. So the genotyping kind of the results was much more reliable and more uniform across the each genes. And so we were very happy with the initial pilot result that we published in the Genome Research, and we rapidly expanded our collaboration together to perform the larger scale single cell genotyping effort. And last year, actually, there have been a simultaneous publication of 2 or 3 papers published about the single cell genotyping effort in acute myeloid leukemia. Our paper in the Nature Communication, and also the paper from Ross Levine group from Sloan Kettering was published in Nature, and also the Stanford group had published the similar effort in AML in the Blood Advances. Because today is my turn, I take the liberty of kind of introducing our own research data, but nonetheless, these 2 or 3 papers really produces almost exactly the same results, and that also speaks to the reproducibility of this single cell platform. So in our study, we essentially performed unbiased kind of large scale high throughput single cell genotyping study. And we had a 123 AML patient bone marrow taken from, mostly at this diagnostic time point, but in some patients, we had a longitudinal time point sample. So in total, we analyzed about 150 bone marrow samples taken from this 123 AML patients. And we used the 2 different targeted panel. One was the 19 gene panel that Mission Bio had developed a priori. And we also designed our own custom panel, which covered 38 genes across using the 290 amplicons. And this kind of large-scale single-cell genotyping study was mainly driven by my graduate student, Dr. Morita, and also bioinformatic analysis was driven by this Dr. Fang, who is also senior computational scientist in our lab. And since when we started this analysis, this Mission Bio platform was a relatively new platform. So we wanted to orthogonally validate all of these mutations by the bulk NGS as well as ddPCR. And also we wanted to validate the copy number data that we get. So we also concurrently run the SNP array for the selected samples. And I'm going to go through some of the QC metrics from this platform, because these are the relatively newer platform that can help your idea of how to use these platforms. So with this platform, with the 150 sample that we analyzed, median about 6,000 cells were genotyped per one run. And sometimes you can genotype more, like 12,000 cells. But depending on the sample quality, sometimes you can only genotype roughly 2,000 or 1,000 cells. But in median, you can get a genotype information of roughly 6,000 cells. And I know with the upgraded version of machine, now you can genotype roughly 10,000 cells with the upgraded platform. And we achieved about 48 depth sequencing depths per amplicon per cell, which we thought is a reasonable sequencing depth because each single cell technically has only 2 alleles. So if you do 48x sequencing depth, you're essentially sequencing 24x per allele in average. And this figure in the right shows this concordance between the variant allele frequency of the mutation that we detected from the bulk sequencing and also the pseudo bulk variant allele frequency that was estimated from the single cell DNA sequencing. And there was a very good concordance, which kind of means that you are only genotyping 6,000 cells from your sample, but that 6,000 is a good representation of the total bulk cells. So we were very relieved to see this, a good concordance between the bulk VAF and also the pseudo bulk variant allele frequency from the single cell analysis. And the limit of detection that we estimated from the dilution assay was about 0.01% of the cellular population. And we also estimated the allele dropout using the commonly mutated polymorphism loci, and that was estimated around 6% in our total samples. And this is a concordance between the mutation detection between the bulk next generation sequencing and also the single cell DNA sequencing. What was gratifying was that 100% of the mutation that were detected by the bulk sequencing, as long as they were covered by your single cell DNA sequencing panel, all of the mutations were also detected by the single cell platform. And actually, single cell DNA sequencing detected a little bit more mutations than the bulk sequencing, mostly in the small variant allele frequency mutation, particularly good for the FLT3 ITD mutation. And this is probably because the bulk sequencing has been notoriously bad on detecting internal tandem duplication for the FLT3. So actually, single cell DNA sequencing platform, because this uses the PCR amplicon-based sequencing, I think it has a better detection or better detection for the FLT3 ITD compared to the bulk sequencing. And all of these small VAF mutations that only the single cell platform detected, we validated these mutations using the ddPCR or the PCR sequencing for the FLT3 ITDs. And when you run your samples with this single cell genotyping platform, you get this type of kind of single cell level mutational heat map. So each cell is lined up in a column, and each mutation is lined up as a row. So here, this is the 1,000 scale that's smashed around here. So in total, for this sample, we genotype roughly about 5,000 cells. And if the cells are mutated, it's coded in the blue. If cells are wild type, it's coded in the white. And if we could not determine the genotype, it is colored in gray. So obviously, you can visualize nicely what mutations are co-occurring in your single cell. Here in these cells, RUNX1 and this NRAS mutations are co-occurring in these cells, but these cells only carry this RUNX1 mutation. And when we run these single cell analysis for these 123 samples, we started to observe this interesting mutational relationship at the single cell level. For example, in this sample, where this sample carried multiple RAS mutation as well as FLT3 mutation, these mutations were actually mutually exclusive at the cellular level. So they were not co-occurring in the same cell, although these mutations are co-occurring in one individual patient. And similar phenomenon was also observed in other cases. For example, in the second case where KIT mutation and the FLT3 ITD mutations were again mutually exclusive at the cellular level, and IDH1 and IDH2 mutation that co-occurred together in this patient were in the cellular level were at mutually exclusive. And this fourth case, which had two different types of FLT3 mutation, one in the tyrosine kinase domain and the other one as an ITD, they were also mutually exclusive at the cellular level. What we gained from these mutually exclusive pattern was that the mutation that have a functional redundancy tends to mutually exclusive at the cellular level, meaning that either you only need one of these oncogenic driver mutation to drive the cancer process, or having two of the functional redundant mutations are actually deleterious to the survival of these cancer cells and therefore negatively selected. And when we summarize this cellular level mutation co-occurrence or the mutual exclusivity patterns, you get a very similar map like the one that you have understanding at the patient level co-occurrence or the mutual exclusive pattern. So the pattern between the patient level observation and the cellular level observation was largely concordant. Although in some cases, we actually found a unique mutational relationship at the cellular level that was actually misled by the patient level information. So actually, we and also a Dana-Farber group had previously performed the bulk sequencing for the therapy-related AML or MDS. And we noticed that these two genes, like p53 gene and also PPM1D gene, are mutated frequently in the therapy-related AML or MDS. And in the bulk sequencing or at the patient population level, these two mutations actually statistically significantly co-occur. And that was a little kind of puzzling when we observed this because both PPM1D and another name is the WIP1 and P53 is working in the same DNA damage pathway. So why does this two mutation that have a similar functional redundancy are co-occurring at the patient or population level from the bulk sequencing study? That was kind of the question we got actually when we did this patient level sequencing. But when we do the single cell sequencing for these P53 and PPM1D co-occurring case, actually at the cellular level, this P53 mutation and PPM1D mutation was mutually exclusive. That is more consistent with the biological functional redundancy of these two mutations. And we actually found about 3 cases of this P53 and PPM1D co-occurrence case that were sequenced by single cell DNA sequencer. And then in all of the cases, we found this mutual exclusivity patterns. So I think sometimes single cellular level mutational co-occurrence and the mutual exclusivity information can correct some of the misleading co-occurrence pattern that is observed at the patient level or the bulk sequencing level. So now that we have this single cell level genotyping map, the next job was to infer the evolutionary history of this tumor development using this single cell map, genotyping map. And why do we want to really understand the evolutionary pattern of these AML development? And this goes to, we were inspired by this effort in the solid tumor field where they had much better understanding about how the pre-cancer to malignant transformation evolves and how the genetic evolution or phenotypic evolution accompanied during those evolution. For example, in the colon cancer, there is a very good understanding of how these benign polyps become malignant invasive cancer. And not only that, they know what is the genetic evolution that takes place during this benign polyp to malignant transformation. And in addition to that knowledge, they also even know how many years, approximate years, that can take from the benign polyp to the malignant transformation occurs. And based on that knowledge, they develop this preventive strategy to recommend the colonoscopy every 10 years in order to prevent these polyps from becoming malignant transformation. Unfortunately, in the acute myeloid leukemia field, we really don't have such knowledge that pre-leukemia phase to the leukemia phase. We recently learned that clonal hematopoiesis can be the pre-leukemic phase, but we yet to know this underlying genetic evolution pattern from the clonal hematopoiesis to acute myeloid leukemia. And we even don't know how many years it might take from the clonal hematopoiesis to acute myeloid leukemia development. So from those perspectives, inferring or understanding the clonal evolution pattern better for acute myeloid leukemia helps our kind of building the strategy for understanding the pre-leukemia to the leukemia process, and as well as the preventive strategy that might be able to be developed. And this task can be also done from the bulk sequencing data, like the one that published in the Nature last year from the UK group, where using the whole genome sequencing data from each cancer types, you can roughly infer the time as well as the relative kind of order of mutational event from the pre-cancer to the cancer development. However, this process is extremely computationally intensive, and also you can kind of get an approximate idea of this linear kind of evolution pattern, but because of this bulk sequencing limitation, you can really not able to understand branching evolutionary pattern from these data. If you have a single cell level genotype data, you actually can get much more granular view of this clonal evolution, particularly this branching evolutionary pattern. To infer this clonal evolution from the single cell genotype data, we collaborated with the group from in ETH Zurich, who had developed this algorithm called CITE, which were designed to infer the evolutionary history from the single cell DNA sequencing data. This was developed from the Professor Björn Winkel and also postdoc of Katarina Jan, and we worked with those groups to build the mutational history map for our 123 AML sample. And using this CITE algorithm, roughly in our total 123 samples, roughly half of the cases had a linear evolution pattern, and the roughly other half had this branching evolutionary pattern like this. In some of the cases with the branching evolutionary pattern showed a very interesting evolutionary pattern, like a convergent evolution pattern, where multiple parallel branches exist, but with the combination of very similar oncogenic function. This has been observed by the multi-site sequencing in the renal cell carcinoma or the pancreatic cancer. However, this was probably the first to observe this phenomenon in acute myeloid leukemia using the single cell genotype data. And now we have built the clonal evolution model and understood the order of these driver mutation acquisition. We try to summarize the mutation order pattern in acute myeloid leukemia, starting from the initiating mutation for acute myeloid leukemia. And the results was not really surprising that most of the initiating mutation for acute myeloid leukemia sample were the one that we know can be observed as a clonal hematopoiesis, reinforcing that these some of these AML starts from these clonal hematopoiesis mutation. However, the mutation that can be acquired after acquiring initiating mutation can be very, very heterogeneous in acute myeloid leukemia. This is a branch only for the DNMT3A branch, but after acquiring DNMT3A mutation, what can be acquired as a second mutation is very, very heterogeneous, although there is some tendency, like in 16% of the times you acquire NPM1, or other 16% of the times you can acquire IDH2. So there is definitely some tendency. However, a lot of pathway is possible for acute myeloid leukemia. And when you summarize all of the possible pathways for acute myeloid development, AML development, or mutational acquisition pattern, the heterogeneity among this mutation order pattern is enormous. Although, and we are currently trying to extract the most statistically meaningful mutation order pattern from these heterogeneous patterns and understand what is the relationship between these mutational order and also the clinical phenotype. And this is because we have been inspired by this effort in myeloproliferative neoplasm, where mutation order have consequence for the clinical, or have a clinical consequence. In this paper in New England Journal, they have described that TET2 mutation first case or the JAK2 mutation first case has a different clinical prognosis or the different clinical features. So we're trying to also replicate these type of understanding in acute myeloid leukemia. Obviously, the mutational complexity in AML is much more greater than the myeloproliferative neoplasm. So getting this clinical correlation at the statistically meaningful way is a little bit challenging. However, we're trying our best to identify this relationship at this point. So far, I have talked about using the single cell DNA sequencing platform to infer the clonal architecture and also the clonal evolutionary history at the genetic level. But these are only the genetic event that we can understand. Now that a single cell platform like this Tapestry platform are able to analyze not only the genetic mutation at the single cell level, but simultaneously analyze the cell surface marker or the cell surface protein expression patterns, we wanted to also expand our effort to understand both the genetic and also the phenotypic evolution pattern. So this is just, this was recently published in the Nature Communication about the technological kind of principle, how we simultaneously analyze the genetic mutation in the cell surface marker using this Tapestry platform. And now this sequencing is called like DAB-seq or the DNA antibody-seq. This is a kind of the example of the data we get from when you run this DNA antibody sequence. Here in the UMAP plot, we are plotting each individual single AML cells as a dot, and you can project the mutation data as well as the cell surface marker information. And it is capable of analyzing roughly 45 cell surface marker at the same time, which is much greater than the actual flow cytometry can do. So we think, you know, connecting this genotype information in the cell surface protein with this scale would have a powerful meaning to understand the clonal architecture and also the clonal evolution history. One of the examples that I show here is that this sample, based on the single cell DNA sequencing results, we knew that this sample had a linear clonal evolution pattern that started with this TET2 mutation and subsequently acquired U2AF1 mutation, followed by DNMT3A mutation and the NRAS mutation. And we projected this cell surface information at this genetic evolution history. And what we learned from this is that from this single mutant cells to double mutant and the triple mutant and the quadruple mutant cell, there was this gradual shift in the phenotype that from the multilineage phenotype to the myeloid phenotype, and then eventually to the phenotype that is similar to the leukemic blast. So projecting both genetic and the phenotypic evolutionary history can tell us that where was the tipping point or where was the transformation point from the pre-leukemia to the malignant transformation. In this case, we predict that the transformation occurred around the NRAS mutation or possibly at the DNMT3A mutational acquisition. So analyzing both genetic mutation and also the phenotypic information through the cell surface marker at the single cell level may be able to help understanding this coevolution pattern of genetic and also the phenotypic evolution. These platforms, I have shown that how these platforms are powerful in understanding the clonal evolution history, both from the genetic side and evolution side. One of the limitations of this platform is that because this platform only analyzes the limited number of genes, in other words, this is a targeted gene panel, single cell panel. And if you use a targeted gene sequencing panel, you lose the information of passenger mutations. You're only profiling the driver mutations at the single cell level. And if you only have a driver gene mutation, although you can understand the mutation order, and if you do the DAB-seq, you can also project the phenotypic shift, you cannot infer the time there. What we mean by that is, like in the colon cancer, going back to this colon cancer analogy, where they have this understanding that pre-malignancy to malignancy takes about 10 years. We cannot really project that information if you only have a driver mutation level of clonal evolution history. We know that the order will be like this, but we don't know how many years for these processes to take if you only analyze the driver mutation. So in order to really understand the time also inside that clonal evolution, you need genome-wide mutation data. Or in other words, you need to also profile all the passenger mutation or an entire somatic mutation at the single cell level. If you have the genome-wide somatic mutation profile at the single cell level using the molecular clock theory, you are able to infer the time also, or absolute timing of the mutational acquisition. And this was recently shown or this effort is kind of driven at the Sanger Institute in UK, where these groups have showed the capability to infer the absolute timing of mutational acquisition in myeloproliferative neoplasm patient. For example, in this bioRxiv paper, where they think this initial JAK2 mutation had occurred 6.2 weeks post-conception, so in utero, essentially. They inferred this based on the mutation rate that we knew from the that happens in the regular HSC, and also the total number of somatic mutation in each single stem cell that they had profiled. In order to understand these absolute mutation timing event, you really need, again, not only to profile the driver mutation, but also the entire genome-wide somatic mutation profile. So in order to do this, we are also trying to replicate this effort that UK group is doing, that perform the genome-wide sequencing profiling at the single cell level. Unfortunately, because of the limitation that I mentioned previously, that amount of DNA is so little in the single cell that without performing whole genome amplification, performing the whole genome sequencing at the single level has been very, very challenging. So the method that we are using is to let the single stem cell or single progenitor cells to create its clone under this mesocellulose culture media, and isolate each stem cell colony and extract the DNA from the colony, assuming that all these colonies have similar genetic background with the original stem cell. And we are performing whole genome sequencing from this extracted DNA from the single HSPC colonies. There are a lot of limitations that I can talk about from this method, but we think this is the best way to at least get the whole genome profiling level at the single stem cell level for clonal hematopoiesis or acute myeloid leukemia sample. So we did this in one of the samples from the clonal hematopoiesis patients taken from the 63-year-old lady who had an 18-year history of treatment for the multiple myeloma and developed this treatment-related clonal hematopoiesis with the mutation of TET2, P53, and PPM1D. And we essentially took her bone marrow and plated the HSPC into this mesocellulose culture and let them grow for roughly 14 days so that the single stem cell creates the colony that is big enough to perform the whole genome sequencing without any amplification process. And there are a bunch of bioinformatic filtering process that takes place in order to come up to the somatic mutation list for this each single stem cells. But in the end, we got this type of phylogeny tree for this patient, which have this parallel evolution of these three different clonal hematopoiesis mutation, P53, TET2, and PPM1D. And based on what we know from the mutation rate at the normal stem cell, and also the years, the number of mutation that is in each of these colonies, we inferred that this original P53 mutation might have occurred 12 years ago from this sampling, which is around roughly close to the time when she started this treatment for the multiple myeloma. We definitely think that we need some revision for this mutation rate analysis for this patient, because this patient was treated with the chemotherapy, so the mutation rate might not be exactly the same as the general population that has been published in Nature recently. But nonetheless, these efforts tell us that this single HSPC colony whole genome sequencing can provide this absolute timing of the mutational acquisition, which is not necessarily inferable if you only perform the single cell targeted gene sequencing, like I just mentioned previously. In addition to inferring the absolute timing of the mutation acquisition, since you have single cell, a genome-wide level mutation data for each single stem cell, you are able to also analyze the copy number profile of each cells, depending on whether it's mutated or the wild type, or the mutation signature analysis, and also obviously the mutate-clonal relationship of the driver mutation information as well. So we think this single HSPC-derived colony sequencing process is a powerful tool to understand not only the mutational history, but also to infer the absolute timing and actual years that take place for the mutation acquisition to happen. And I think we are building these effort and the cases in order to make this publishable. Lastly, I'm going to talk a little bit about the implication of these clonal heterogeneity and evolution pattern in the AML treatment. So even with the bulk sequencing era, we knew from these beautiful paper from the WashU that if you perform the whole exome sequencing at multi-time points at the diagnosis, after the treatment, and also at the time of relapse, you can understand this very nice clonal evolution pattern that occurs during the acute myeloid leukemia treatment, where AML chemotherapy provide a strong bottleneck for the clonal selection and the resistant clone that gets selected under the treatment and causes the relapse. If you perform the single cell DNA sequencing at multi-time point samples during this AML treatment, you get much more granular view of how these individual clone behaves during this AML treatment. These two examples were taken from the AML patients who were actually primary refractory to the treatment that we were giving. So when you only look at the bone marrow at the histological level, you start with the folloblast, you ended up with very similar type of picture. So you think, okay, the treatment didn't work. There's not much to learn from these cases. But if you actually look these cases with the single cell DNA sequencing, there's a very complicated clonal dynamics occurring, even though as a net effect, the patient is not responding to the treatment. So in these two examples, we are showing that some individuals' clones are being treated with your treatment, while the other clone is selected or emerging during the treatment, which essentially caused the primary refractoriness to these patients. There are lots of examples like this, which we have shown in the papers, not only the primary refractory case, but also from the diagnosis to relapse case. And I would like ask you to read those papers to get more patterns from the clonal evolution. But nonetheless, I think the single cell DNA sequencing data provides really the granular view of individual clone level response to the treatment, not at the patient level, but now individual clone level response to the treatment. And this has been applied to the multiple context now in the FLT3 inhibitor treatment context or IDH inhibitor context, and also the BCL2 inhibitor context. And what we are observing from these efforts for the targeted therapies context is that one of the kind of uniform characteristics of the resistant mechanism is that mutations with the RAS or the clone with the RAS mutation or the receptor tyrosine kinase mutation tends to be selected with these targeted agent treatment. And this was also nicely described from the Craig's lab, where they not only look at the genetic evolution pattern during the venetoclax treatment, but simultaneously analyzing the phenotype and kind of inferring that actually not only that the RAS mutant clones are the resistant, but it could be that the monocytic differentiation state could be the mechanism of the resistance to the venetoclax treatment. I think this monocyte or the RAS relationship is kind of the chicken and egg relationship where, RAS mutant clones are frequently monocytic, and also monocytes tends to carry the RAS mutation. So we don't really know at this point which one is the primary driver of the resistance, but I think nonetheless, profiling both the genetic and also the differentiation state provides a more mechanistic view of what is driving the resistance against these targeted therapies. And this multi-omics capability is really kind of evolving in this field. I showed the capability of simultaneously profiling the DNA and also cell surface marker at the same time. But there is also lots of capability like RNA sequencing as well as cell surface markers, and also potentially analyzing DNA and RNA together. And I think we are in really the exciting time to observe this evolution of how the single cell technology is really rapidly evolving. So as a summary, application of emerging technologies like single cell technology started to reveal remarkable clonal heterogeneity in acute myeloid leukemia, which is enabling the better understanding of mutation order and also the history. I also touched base that not only profiling the driver mutation is probably not enough to understand the absolute timing of the mutation event, and we are trying to also infer those things through the genome-wide sequence, a whole genome sequencing of the single stem cell level. Clonal heterogeneity seems to be the main driver for the resistance of the targeted therapy in AML, and multi-pronged evaluation of cell state, not only profiling the genetic state, but the multi-dimensional analysis, DNA, RNA, or DNA protein, RNA protein, those will shed light on the most important mechanistic insights into the drug resistance in acute myeloid leukemia. With that, I would like to really thank my mentor and also my lab people, as well as all the funding source, and also the outside collaborator, especially ETH Zurich people, and also Mission Bio people also provided lots of technical assistance when we really performed this large scale analysis. So we really wanted to thank them too. With that, I would like to end here and happy to answer any question if there is.
Koichi, thank you for a wonderful seminar. That was even more compelling than last time I heard it. So let me start by asking you a couple of questions, if I may. So when you observe this very dynamic sort of clonal evolution for patients on therapy, is that what you always see? Is that the prevalent sort of dynamic, or do some patients less so? Or what's your experience with that? I'm just curious.
Yeah, actually, we did cherry pick some of the interesting case for the publication as well as these slides. So yes, actually, majority of the case, sometimes it's the clonal evolution pattern is boring. In other words, that there's really no genetic changes. But we kind of see that these dynamics more with the targeted therapy or low intensity treatment context. So if you use very intensive chemotherapy, they are pretty good about addressing these clonal heterogeneity. So they're really good at eliminating lots of small clones and only selecting certain clones. But in the targeted treatment or the low intensity treatment, I feel like we are seeing more of these complicated clonal dynamics taking place, which results in the selection of these resistant clones.
Wow. And you mentioned that about half of the samples you looked at showed branching evolutionary patterns versus linear. So what Shanshan has gone on to do from the data you mentioned is to identify independent leukemia stem cells, genetically independent leukemia stem cells within the same patient. So based on what you told us today, I would imagine those are the branching clonal evolution patients where you can evolve to more than one leukemia stem cell subtype, which says that can happen in roughly half the patients based on your numbers, which I think is fascinating. Anyway, let me let other people ask a question.
No, actually, that was very, very good question. And we actually, in our paper, we had a small section where we transplanted these branching evolution sample into PDX mouse and performed a single cell DNA sequencing of engrafted samples. And I have to say that we only were able to profile the one passage, not the multiple passage when we published, but at least after the first passage, we saw that every branching subclone was engrafted, and we were able to see that being engrafted. So we think that each of these branching subclone have leukemia initiating capacity. In other words, I know that it doesn't mean that they have a leukemia stem cell capability, maybe per se, but at least leukemia initiating capability in this PDX model. So we got very interested in about these kind of multiple parallel leukemia initiating capabilities.
So the trick that John Dick uses is just to go to limiting dilution to those samples, right? So sooner or later you'll separate out the individual's stem cell population.
Right, exactly.
Mercedes, did you have a question? You're still on mute.
Sorry. So I was wondering, from the beginning of your talk, when you find out that there are two mutations that you say could be redundant, but then when you did single cell, you find out where so I don't know if you of the field have looked at something similar to where in the SNPs it's called the linkage disequilibrium, but in terms of mutation. So meaning, say with the SNPs, you need two SNPs completely different locus to have a phenotype, and if you have only one will work. So could it be possible that you need something similar, but with different cells? So basically, one cell has a mutation in P53, but that will not have phenotype. At least the other cell that could help have a mutation in something else. So similar concept to the linkage disequilibrium, but in the context of different event cells, it's not different locus.
Wow, that's really fascinating point, and I never thought about in that way, but I think that is extremely interesting point. And since these platforms are targeted sequencing platforms, it might be a little bit difficult to screen for these SNPs, but if we can expand the genome coverage, that is definitely possible. And I think maybe interesting results might come up from that, actually.
Mm-hmm.
When we observe these, functional redundant mutation being mutually exclusive, we actually search a lot of literature if someone have done this in the cell-based model. And we were actually surprised that there were not a lot of papers that tried to, let's say, put the two NRAS mutation in cells and analyze the kind of advantage of those cells. We found like a few papers, one from Sloan Kettering, where they used the lung cancer cell line and put two different EGFR mutation. And that was actually, they're showing that was deleterious to the cell survival. So we cited that in our paper, but I actually never had some way of thinking that like maybe germline SNP could actually play a role here. And I think that is extremely interesting.
Yeah, or even culture, for instance, culture in cells with one mutation with cells with the other mutation together.
Right.
See whether that can actually help each other.
Yeah, that's very, very interesting point. I have not done that, or I've never thought about that way. So we never tried, but that's very inspiring, actually.
Thank you.
Eric, let me go to you in one moment. I just wanted to mention to people, if you're shy about speaking, put your question in the chat box. I'm happy to read it for you. Eric, you had a question?
Yeah, first off, absolutely fascinating talk. And, a couple of implications I was thinking of in terms of the data you present is, in esophagus and many other tissues, we've begun to see clonal evolution in the standpoint of how non-malignant clones that carry mutations essentially increase fitness and potentially could provide competition against clones that are malignant. And I'm curious, a lot of your analyses are focusing on basically the evolution of the malignant clone, but do you see any particular evidence for, say, clonal competition between non-malignant clones and malignant clones where you start to see certain mutational spectra are favored in competition with AML, or that, lead not to AML, but, simply expand well alongside it?
That's also a wonderful question. If you only profile the DNA, I think separating the non-leukemic versus leukemic cell was a little bit difficult to do, although we saw several examples where we saw like a bystander non-leukemic, likely bystander non-leukemic clone just from the pattern of the mutation. But I think if we use this, cell surface marker plus this DNA analysis more and more, we can probably tease out a lot of this question that in AML ecosystem, that actually it's not only the leukemia cell, but there are bystander non-leukemic clone, and whether those are affecting the fitness of AML cells or not. I think those are extremely interesting questions. We have observed these bystander clone at the genetic level, but we haven't yet analyzed whether that is affecting the fitness of these leukemic clone. I think in the clonal hematopoiesis field, at the population level, if you have a multiple clone, that increases the risk of transformation. Maybe it's just because you have a multiple clone in place, so you have a better, have a higher chance of it transforming. But whether these multiple clones are competing each other and what is causing that, I don't think we have a good understanding on that yet.
Yeah, maybe a great thing even just to have a longer chat about. Just, I'd love to get some of your insights into what you might be seeing.
Yeah, that's extremely good question. Yeah.
Other questions for Dr. Takahashi? I know he has a session with a group of trainees after this, so you can ask your questions there as well. Shweta, did you want to ask a question?
Yes. So I had a question. So you're trying to see if there's a way to predict at the time points of when mutations can happen and a way of prevention. Do you think it is also possible to predict if the patient is going to go through a branched evolutionary pattern or a linear evolutionary pattern and give a treatment accordingly?
Yeah, we also try to kind of identify the predictor for the linear and the branching evolution. And when we compare the clinical feature between the two patterns, unfortunately, in our case, we didn't find anything that stood out. And these linear pattern and the branching pattern is in somewhat a little bit misleading too, because since we are only profiling a limited number of targeted genes, if you expand your genome coverage, I'm pretty sure every case have some degree of branching evolution there. So it is linear in a sense that you only profile certain targeted genes, but I don't think there is a pure linear evolution pattern in AML actually. So probably all of the cases have a branching evolution pattern. It's just, you need to widen your genome coverage in order to understand that. So at this driver mutation level, we didn't really identify the predictor for the branching and the linear. Maybe a little bit similar, but a little bit different one is that we at least analyzed the correlation between the number of subclones and the clinical feature, and older patients tended to have more number of subclones, which is very intuitive that, in order to have a heterogeneity, you need longer time to, develop. So I think older patients, AML tends to have multiple clones. That doesn't necessarily mean that these are linear or branching, so.
Yeah. Thank you.
Oh, sorry.
Oh, I was just saying thank you.
It'd be fascinating actually if things like induction chemo were triggers for switching to a branching mode. So I think we're cutting into the time of the trainees that we're going to meet with you, Koichi. So I'm going to wrap it up now. But again, wanted to thank you for a really fascinating seminar.
Thank you very much for the opportunity.
Thank you.
Thanks for listening.
Bye-bye.
Have a great day, everyone. Bye-bye.
Bye.
So trainees can stay on the call if you are signed up to meet with Dr. Takahashi.
There we go. Thank you, Adela. So don't go away to Koichi.
Yes, no, I'm not going.
I'll follow up with you soon. Bye-bye.